Hey all, in this first video on regulation of gene expression, what we're going to talk about are simple bacterial models of gene expression that are known as operons. So let's talk a little bit about the structure of an operon. An operon is a DNA sequence within prokaryotes that is required to control the expression of genes. Though we're looking at prokaryotes here, what we find is that this serves as a great model for what's occurring within eukaryotes as well. So let's take a look at the basic structure. In blue here, I'm showing you what's known as the promoter. You may recall that the promoter is specifically where RNA polymerase is going to bind to DNA. And we've actually got two promoter regions here. Just downstream of the leftmost promoter, we have what's known as a regulatory gene. This regulatory gene controls whether the rest of the operon is going to be turned on or off in the process of transcription. Oftentimes, these regulatory genes bind to what's known as the operator, and you'll notice that the operator is downstream of that second promoter there. We may also find additional control elements where transcription factors or activators and repressors may bind in order to activate or repress gene transcription. And finally, we have our genes that are being regulated by this full operon. Now, one last thing to note here is that these arrows that I'm showing right downstream of the promoters are what's known as the transcriptional start sites. These transcriptional start sites are where the first nucleotide of mRNA will be produced during the process of transcription. So here is the general structure of the lac operon. Just like the image we saw, we have a couple of promoters. We have now, instead of a regulatory gene, the LAC-I gene, which functions as that regulatory gene. Instead of an operator, we have LAC-O. Instead of genes, we have the specific genes LAC-Z, Y, and A. And instead of a control element, we have the cap binding site. One of the things that's going to happen first is that the RNA polymerase with what we call a sigma factor is going to bind to the promoter. And notice how the sigma factor is helping to place the RNA polymerase directly over the transcriptional start site. Well, the sigma factor does this by binding to two sites in the promoter that are known as the -35 and -10 sequences. This is a very specific sequence of DNA that we find in the promoter of not only this gene, but many others as well. And once this happens, the RNA polymerase is then going to proceed, and it's going to end up transcribing the LAC-I gene, which then is translated into the LAC-I protein. Now, one of the things that we should note about this is that this process is regulated constitutively. In other words, what we mean is that this LAC-I gene is always being expressed. That's in comparison, or a little bit different than what we find in the rest of the operon, which the gene expression is regulated, meaning it's going to be controlled by the binding of proteins to that cap binding site, the promoter, and the LAC-O, or that operator site. Now, the LAC inhibitor, or this LAC repressor, binds to DNA sites specifically, and it binds to a specific sequence found in the LAC operator, or the LAC-O sequence. And you'll notice that this is occurring between the transcriptional start site and the promoter. So, if the RNA polymerase were to try and bind, the LAC inhibitor is going to prevent it from proceeding to transcribe these genes known as LAC-Z, LAC-Y, and LAC-A. In essence, this functions kind of like a light switch. When LAC-I is present, transcription is repressed or turned off. But when LAC-I is removed from the DNA, transcription is turned on. And this can toggle between the on and off state depending on whether LAC-I is bound to the LAC operator. Now, how is this LAC operon then regulated? How is it ever turned on? Well, we just said that this happens in cases where the LAC inhibitor no longer binds to the DNA. In fact, that inhibitor is allosterically regulated. So, for example, when we have lactose around in a cell, like so, the LAC inhibitor, which is shown in orange here again, will unbind the DNA when lactose binds to it. And it will change shape. And the way that it changes shape is such that it no longer can bind to the operator. In cases like this, the RNA polymerase can now bind to the promoter and activate transcription. Now, while the LAC inhibitor may be the on-off switch, there's another protein that functions as the dimmer switch, and it can increase the intensity in which these genes, the LAC-Z, LAC-Y, and LAC-A, are being transcribed. That protein is an activator, and it's known as CAP. The A here in CAP stands for activator, and this protein will bind to the CAP binding site when it's bound to ATP. Now, whereas the LAC inhibitor may function as the on-off switch, there's another protein that functions like the dimmer, and it can increase and decrease the intensity in which the LAC-Z, LAC-Y, and LAC-A genes are being transcribed. This protein binds to the CAP binding site, and this protein is known as CAP. And in fact, the A in CAP here stands for activator, and so this is an activator of transcription. Well, CAP only binds to the DNA when it's bound to a molecule known as cyclic AMP. Cyclic AMP is kind of like a brethren of ATP, but it is produced in times or in conditions when energy levels in a cell are very low. So let's imagine here again in this scenario where we have this LAC inhibitor. And when LAC inhibitor is bound to DNA, the transcription of the LAC operon is off. However, if lactose is added, the LAC inhibitor changes shape when it's bound to lactose, and that's going to turn it into an on state. And the loss of lactose then turns it back into an off state. Well, let's not forget that there's a second level of regulation here, and that second level of regulation here involves the protein CAP, which binds to cyclic AMP. And whereas the LAC inhibitor is the on-off switch, the CAP protein will take some transcription and turn it into lots of transcription by recruiting the RNA polymerase or activating the RNA polymerase at the promoter site.